And welcome everybody again to our second regional anatomy tutoring session. We don't have too many announcements because we just saw you guys last week. Just a couple of things to note. One is that we just sent out the announcements about the recipients of the ABITS sponsorship winners. So if you did not receive an email and you did apply, please contact us and we'll make sure that you get your notification either way. That being said, if you did not receive funding, we strongly encourage you to reach out to your high school or your community to see what sponsorship they might be willing to offer. We at the Anato-Bee are so willing to provide any letters or endorsement that we can to help you get that funding from them if need be. So reach out to us if you need any help in writing that letter, getting that to them. Another thing from Dr. Peterson and some amazing students at Drexel, they have put together a short video for you on instructions for what to do in the cadaver lab. So that should help you for the practical, kind of giving you some tips on how to get oriented, some good tips and strategies from some medical students that have been through the anatomy lab practicals quite a few times at that point. And we'll have that up on the YouTube channel and onto our website this week. So looking forward to that. And thank you, Dr. Peterson, and to your students. Final update that I have is that the next tutoring session has been moved. It is the review session that we have scheduled because we did not realize that it was scheduled for Easter Sunday, and that would probably limit a lot of attendance to what is supposed to be an interactive tutoring session. So we've moved that to a Thursday evening. It should be March 28th, so still giving you quite a bit of time in advance before the competition to get those questions in. Very similar structure to that of what we're doing for the local competition, but now with this added layer on of the these standardized patients and the oral examination. So we'll have some examples in there for you too to give you a taste of what that will look like. So any other things that I'm missing, Dr. Peterson? I think we covered it. All right. So with that, I would like to introduce our first speaker. That is going to be Dr. Whitney Carriger. She is an assistant professor of anatomy and the anatomy lab director at the Edward Via College of Osteopathic Medicine in the Auburn campus. She has a total of 4 degrees in anthropology, and that is the study of humanity or understanding human behaviors, biology, cultures, societies, basically anything and everything that makes us human. So she earned her bachelor's degree from the University of Michigan. She then earned 2 masters in anthropology, one from Illinois State University and another from Tulane University. She then continued on to receive her doctorate from Tulane University. And Dr. Carriger taught at the Alabama College of Osteopathic Medicine for 2 years before joining our VCOM family. In my time working with her here, I can say she does an amazing job of breaking down complex topics, and all the students just love her for the care that she puts into their learning. And just obligatory, have to put the pets in there. Dr. Carriger does have 2. She's got a dog named Rudy, super sweet, and a cat named Mac. So without further ado, I'll let Dr. Carriger teach us all about cardiopulmonary system.
And thank you so much. I just want to thank the organizers of the Anato-Bee for inviting me to do this. So I'm really grateful for this opportunity. I'm going to get my screen shared. Okay, can you see the screen okay, everybody? Okay, cool. I'm going to move this out of the way. Okay. So I'm going to be reviewing the cardiopulmonary system today. So I know that you all have had some tutoring sessions on this already. So some of it will just be kind of review from things that you've heard in other tutoring sessions. And then I noticed that some of the learning objectives were new. So some of it might be a little bit of new material too. But I'm going to try to cover just a general overview of heart and lung anatomy, embryology, and histology. Oops, and I can't seem to get, I need to do this. Okay, can you see the laser pointer, hopefully? Okay, cool. Okay, so I sort of divided the learning objectives up for anatomy first, and then histology, and then embryology. So I have like an overview slide, but then basically the title that's listed on each slide will sort of fit with 1 or 2 of the learning objectives. So, but we have this overview here, if you wanted to go back and look at what the learning objectives were. So just as a reminder, our heart and lungs are important for breathing and for pumping oxygenated blood throughout our body. So of course we're going to breathe in, get oxygen into our lungs, and our heart is going to be sending oxygenated blood out to our body, and then veins will carry that deoxygenated blood back from our body back to our heart, and then our heart will also pump that deoxygenated blood out to our lungs to get oxygenated again. Then the oxygenated blood will go back out to the heart, and then it'll continue that cycle out to the body. And so these are just some sort of overview images of the heart and lung functioning. And I know that you probably covered this already in previous sessions, but I realized there wasn't really a learning objective for it. So I just wanted this introductory sort of overview slide. So the organs that we're going to be talking about today are in our thorax. And so we can divide our thorax into 3 compartments. And so first we're going to talk about the compartment that's in the middle. So we have 3 compartments. 2 of those are the left and the right pleural cavities, and that's where our lungs will be. And we'll talk about that in just a little bit. But first we're going to talk about this 3rd compartment that's in the middle here, called the mediastinum. And so that's going to include our heart and some other structures too that I'll talk about in just a minute. First, we can divide our mediastinum into 2 parts. So we can divide it into a superior part, which is above this green line. So I guess it's just this piece here. So above the green line here in the image, and also above this green line, and then an inferior part down here below the green line. And so this green line is placed at about the angle or the level of the sternal angle. It was a little bit hard, of course, in this one because we can't see the sternum. But that is about where the division of the superior and inferior mediastinum is, at the level of the sternal angle. And so here's the superior mediastinum, as I already mentioned, and then we have the inferior. And then we can also divide the inferior mediastinum into 3 parts. And so this view up here, the sagittal view up here, helps us see those different parts. And so I've color-coded them. So the sort of orange part here in the front, that would be the anterior part of the inferior mediastinum, and then a middle part here in blue, and then this yellow part here posteriorly is the posterior part of the inferior mediastinum. Now for the contents, we have in the superior mediastinum, we're going to see the aortic arch here and some of its branches. We're also going to see the superior vena cava and tributaries to that superior vena cava. We're also going to have some nerves running through that superior mediastinum, the trachea, which we can see a little bit back here, the esophagus, which we can't see in this image because it would be located posteriorly to the trachea, and then also part of the lymphatic system, the thoracic duct. And then down here in the inferior part of the mediastinum, anteriorly, we're going to have the thymus and some lymph nodes, and that will really, the anterior part is the part between the front part of the pericardium, or the sac that surrounds the heart, and then posteriorly to the ribs and the sternum out at the front of the thorax. Then in the middle of the inferior mediastinum, we're going to have the heart, the pericardium, the roots of the great vessels, some other arteries, and the phrenic nerve as well. And then posteriorly here, this posterior part of the inferior mediastinum will run again from that posterior part of the pericardium, which is that sac surrounding the heart, back to the thoracic vertebrae. So these are the thoracic vertebrae out here. So all of this yellow is posterior, the posterior part of the inferior mediastinum. And in there, we're going to find the descending aorta, the azygous, hemiazygous, and accessory hemiazygous veins, the thoracic duct, which is part of that lymph system, the esophagus, and I guess I already mentioned veins and lymphatics back here in this posterior part. Now I mentioned the pericardium in the previous slide, and so that is sort of a sac surrounding the heart. So we can think about the pericardium as being sort of a balloon, and then if we think of this fist here as the heart, and imagine that sort of just pushing down into the balloon and creating sort of a double-walled sac here surrounding the heart, that's how we can think about the pericardium. So the pericardium is going to have a fibrous outer part. So what this image down here is showing us is this is part of the heart muscle, and then all of these are going to be parts of that pericardium. So that's what's being shown out here. So out here we have the fibrous part of the pericardium, that's sort of the most external part of the sac surrounding the heart. And then we're going to have a serous pericardium. And so that can be divided into two layers. A parietal pericardium. So parietal is usually something in anatomy referring to something that lines the wall, or like the parietal bones of the skull, or sort of like the walls of the skull. So the parietal pericardium is going to be this layer that's out here, sort of stuck on the inner surface of the fibrous pericardium. And then the visceral pericardium, in this case, visceral is referring to the organ. And so the visceral pericardium will be lining that external surface of the heart. And then in between the two, there's going to be a space with a little bit of serous fluid in it. And that's really important for cushioning the heart and for reducing friction when the heart is beating. Okay. So the next learning objective then is a little bit about the heart conduction system. So we have two sort of important nodes that we have to think about when we're thinking about the heart conduction system. So we have the SA node and the AV node. The SA node is that's the node that sort of establishes normal cardiac rhythm, and you can think of it as like the pacemaker of the heart. So that's located here in the right. So this is the right atrium. Right ventricle, left atrium. So this is the right atrium here, and this yellow dot up here at the top, sort of by the superior vena cava, that is going to be the AV node. So it's located sort of in the more superior part of the right atrium near that opening to the superior vena cava. And then the AV node, that's located down here, more inferiorly, also in the right atrium though, but down here more inferiorly. So the AV node is going to get impulses from this SA node. That's what this image is showing. And then the AV node is going to pass them to the AV bundle. And then those impulses are going to be transmitted down this interventricular septum here and out to the ventricles. And so this, of course, this system of this heart conduction system is what's making our heart beat. And so that's important for pumping all of that blood that we were talking about, both out to the lungs to get oxygenated, but also then out to the body once it's been oxygenated. And then in terms of what's increasing and decreasing our heart rate, it's the sympathetic or the parasympathetic nervous system that's decreasing our heart rate. So when we think of our parasympathetic nervous system, that's usually having to do with like rest and digest things, whereas our sympathetic nervous system is more to do with like fight or flight things. So if you're in some sort of stressful situation, your sympathetic nervous system is going to sort of take over, and at least in terms of heart rate, that'll be what increases the heart rate, and the parasympathetic nervous system is going to be what decreases the heart rate. Okay, and then in terms of anastomosis, so I know that you probably all learned about various arteries throughout the body, and we often will think of arteries in terms of how blood is flowing from the heart out to different parts of the body and sort of make sense of these pathways of blood flow in our head. But we have a lot of these anastomoses in our body, and so we can think of these as sort of interconnections between different arteries. And these are really important because they create redundancy in blood supply to certain areas. So, and that's important because then if one part of that flow system is blocked, there can be other pathways for blood to get through to get out to your tissues. So, we have some anastomoses around joints, for example, and that's really important. If you have a joint bent, you'll be compressing probably at least part of those blood vessels surrounding that joint. But the anastomoses around the joint can still allow blood to flow past that joint. Another really important system of anastomoses is in our brain. Of course, the blood supply to our brain is really important. And so we have this circle of Willis here in the brain, and that is sort of a way for blood to get to different parts of the brain following more than one pathway. Okay, so that was sort of the heart learning objectives for anatomy. So now we will move on to the lung learning objectives for anatomy. We already mentioned how the thorax can be divided into 3 compartments, and we just talked about the mediastinum, which is in the middle, which in this image here, it's just empty because the heart's been removed because we're focusing on the lungs now. So We talked about the mediastinum, so now we're going to talk about the other two parts of that thorax, which are the left and right pleural cavities. So we have the left pleural cavity here, the right pleural cavity here, and these are each going to contain one of your lungs. Just like with that pericardium and the heart, we're going to have pleura lining the lungs and lining the cavities. So we'll have parietal pleura. Remember, I said that was usually having to do like with a wall, for example. So we have parietal pleura lining the walls of these pleural cavities. And then we're going to have visceral pleura, again, having to do with the organs, so lining the surface of the heart. And just like with the pericardium, there will be some space between that parietal and the visceral pleura, and it will contain a little bit of pleural fluid. And that's really important for reducing friction during breathing. But it's also really important because that fluid in there sort of keeps the layers of the pleura stuck together, and that's going to help the lungs expand and fill with air when you breathe in. So as a review, our right lung, that's going to be a little bit bigger usually than our left lung, and that's because we have the heart here in the mediastinum, which is usually offset a little bit over to the left side. So our right lung will have 3 lobes, a superior lobe, a middle lobe, and an inferior lobe. And those lobes are going to be separated by 2 fissures. And then the left lung will have 2 lobes, a superior and inferior, separated by just 1 fissure. If we were to look at the hilum of the lung, which is, if you can imagine, sort of cutting right here and turning the lung so that we're looking at that sort of medial surface of it, the structures at that hilum of the lung will be in a different order, so we can identify lungs that way too. So at the hilum of the lung, on the right side, the pulmonary arteries are going to be located anterior to this primary bronchus. So this is the trachea. That'll split into a left and a left and a right primary bronchus. So the arteries will be anterior, the pulmonary arteries coming from the heart, they'll be anterior to this primary bronchus on the right side, and then superior to it over here on the left side. Just a couple other important things is that it's our phrenic nerve that supplies the diaphragm. So our phrenic nerve is really important for allowing us to breathe, because that's what's supplying the diaphragm, which is a sort of a muscular layer that would be sitting, you could imagine a muscular layer sitting right here under the lungs, separating this thorax from the abdomen that would be below it. And then we also are going to have some sensation supply, some sensory supply to our trachea and our bronchi. So I mentioned the trachea and the primary bronchi on the previous slide. So in this image here, this is representing the trachea right here. So when we're breathing, air is coming in like through our nose and mouth, going down to our trachea, and then the trachea will divide into a left and a right primary bronchus, one going to each lung. And then on both sides, then the primary bronchi are going to split into secondary bronchi. And I'm sorry, this 3 and the 2, that should be with the secondary bronchi. So we'll have 3 secondary bronchi on the right and 2 on the left. It's a mistake on the side. And then those secondary bronchi will split into tertiary bronchi. From there, we go out to bronchioles. And so we have some different kinds of bronchioles. We have terminal bronchioles, or conducting bronchioles, and then terminal bronchioles, and then respiratory bronchioles. And then after that, we have alveolar ducts and sacs. And so we can sort of divide all of this branching of this tracheobronchial tree that's getting air from outside our bodies into our lungs. We can divide it into 2 parts: the conducting system, which is everything to the terminal bronchioles. So all of this system up into the terminal bronchioles, that's all considered part of the conducting portion of the respiratory system. And that's because its function is really just moving air. So it's just moving air into and out of our lungs. And then the respiratory bronchioles out to the alveoli. That's the respiratory portion of the tracheobronchial system. And that's because that's where gas exchange is happening. So the air is moving through all of this part, and then once it's getting out to the respiratory bronchioles and the alveoli, that's where gas exchange is happening so that we can get oxygen into our bodies. Okay, so now we are going to move on to the histology. So we'll do artery and vein histology first, and then the respiratory histology. Okay, so in arteries and veins, we can think of them all as having sort of 3 layers. So they're all tubes, and we can think of each of those tubes as having 3 layers. And those are called the tunica intima, which is the most internal layer. Over here, it's being represented right here. And then the tunica media, that's the middle layer, which is right here in this image. And then the tunica adventitia, which is the most external layer. So if this was, I know it's not a complete circle, but if it was a complete circle, blood would be flowing through this area out here, and then out here would be sort of outside this artery. So we have an artery here and a vein here. And you can see even in this vein, we also have those 3 layers. So the tunica intima on the inside, the media in the middle, and then the externa or adventitia out here on the outside. And arteries and veins are both going to be classified based on size. So we can think of, so we're going to have large or elastic arteries, medium or muscular arteries, and then small arteries and arterioles. And then when we get to veins, well, you're going to have large veins, medium veins, small veins, and venules. And then the capillaries are sort of that transition part in between the veins and the arteries. So the arteries will carry oxygenated blood out, and the veins will carry deoxygenated blood back, and sort of that transition area is the capillaries. Now, in general, arteries are going to have a thicker wall relative to the size of the lumen. And because they have this thicker wall, in histology slides, oftentimes these arteries will stay more rounded, at least compared to the veins. So this is a vein up here, and it still has a pretty thick wall, but it has a really big lumen. So I guess the wall size, the wall thickness compared to the lumen, we have a much thinner wall compared to a bigger lumen here in veins. And then in arteries, we're going to have a thicker wall and a smaller lumen. And then, so I have pictures of these on the next 2 slides to show you. And I'm sorry if you can hear my dog barking in the background, but so the sort of the distinguishing feature between large and medium arteries is that the medium arteries are going to have a clearer internal and external elastic lamina. And so the internal part is going to be what separates the intima from the media, and then the external part separates the media from the adventitia. And so this is present in the large ones, but it's not really very clear in histology slides, but we can see it a lot better in the medium ones. And then these smaller ones here, they will have a lot less smooth muscle in them. These small arteries and the arterioles will have less smooth muscle. Okay, so this is an example of a large artery over here, or an elastic artery. And I included these because I liked that it had these drawings sort of labeled. So we have the tunica intima here, and this would be the intima, and then the media, and then the adventitia. So this is an elastic artery compared to a muscular artery over here. And so this internal elastic membrane especially is easy to see. And so the presence of this internal elastic membrane and the fact that we can see it really well is sort of what can help us distinguish this muscular artery from this elastic artery over here. And then when we go to the next slide, we have, this is an example of a vein. So it shows an artery. So this is also an artery. This is an artery, but here's the vein. And again, we can see that there's a thinner wall, at least relative to this much larger lumen in the vein. And then this image over here shows a venule and an arteriole. And again, we can tell the arteriole apart from the venule because the arteriole is going to have still a thicker wall compared to the smaller lumen when we compare it to the venule. And then over here, we have capillaries being shown within adipose tissue. Oops, my slides won't change. There we go. Okay, so now we're on to histology of the respiratory system. And again, we start with the trachea, and then that'll branch into primary, and then secondary, and then tertiary bronchi. Those will branch into bronchioles, and then alveoli. So what distinguishes the trachea and the larger bronchi from the smaller passageways are that they will have, well, the trachea has C-shaped cartilage rings. And then when we get out to the primary bronchi, you'll have complete cartilage rings, sort of supporting those bronchi to keep them open. Once we get out to the terminal bronchioles and respiratory bronchioles on out, there won't be any cartilage rings left. So the lack of cartilage can help sort of clue you into it being one of these smaller structures here. And the larger structures will have the presence of cartilage in them. And then another thing we can look at is the kind of epithelium. So our trachea and our bronchi will have pseudostratified columnar epithelium. But once we get out to the terminal bronchioles, we'll see some low columnar or cuboidal epithelium. Also, we can see cuboidal epithelium in the respiratory bronchioles. And then once we get out to the alveolar ducts and alveoli, we see simple squamous epithelium. So this is a picture of the trachea. This is also a bronchus. And then these are bronchi images here. And what I really wanted to point out here is that we can see some hyaline cartilage, because remember, in the bronchi we have, and in the trachea, we have cartilage sort of keeping those airways open. And we also have that columnar epithelium. When we get out to the bronchioles, remember we have more cuboidal epithelium. And this is a transition from a bronchiole here, a terminal bronchiole, out to a respiratory bronchiole. And then when we get out to the alveolar ducts and the alveolar sacs, we're going to have more simple squamous epithelium. So that's how we can identify that. Okay, so now we are on to the embryology. First, we're going to do part embryology. So this is the first functional organ to develop, and it's going to develop from mesoderm and the cardiogenic area near the head of the embryo. So that's what's being shown up here. And so the cardiogenic cords are going to develop into sort of tubes with a lumen or a hole in the middle. Those tubes will eventually fuse and then sort of divide into different parts. And so these different parts here will eventually give rise to the parts of the adult heart. So the truncus arteriosus will give rise to the ascending aorta and pulmonary trunk. The bulbus cordis will give rise to the right ventricle. This primitive ventricle will become the left ventricle. And then the primitive atrium will become part of the right and left ventricle, or I mean right and left atria, I'm sorry, and auricles. And then the sinus venosus will also become part of the right atrium, the SA node, and the coronary sinus. And so basically this tube will elongate, it will eventually fold, and then the septa and the valves will develop and the chambers will remodel into the adult part that you know about. There are some differences between fetal and adult circulation because, of course, a fetus isn't breathing, right? It's getting oxygenated blood from its mom. So its lungs aren't functional. It isn't breathing. So it can't have the same sort of pathway of blood flow out to the lungs to get oxygenated and then back to the heart to go out to the body that adults have. So we have these three structures here that sort of help to change that pattern of blood flow. So we have first something called the foramen ovale. This is an opening in the interatrial septum, so between the right and the left atria. And this allows blood to flow from the right to the left atrium. And again, that is helping the blood sort of bypass going out to the lungs and then back to the heart. And after birth, once this closes, and it will become a structure called the fossa ovalis. Another structure that sort of helped diverting blood away from the fetal lungs is the ductus arteriosus. And this is going to connect the pulmonary trunk and the aorta. Again, at birth, this will close up and it's going to become the ligamentum arteriosum. But of course, we need oxygenated blood to get to that fetus still. And so how that's happening is through this ductus venosus, which is a branch of the umbilical vein. And that's allowing oxygenated blood in the placenta to go to the heart of the fetus. And then this will close more slowly during infancy and eventually become a structure known as the ligamentum venosum. Okay, we're almost done. So now we're on to lung embryology. So the lung bud is going to sort of branch out from the trachea, and that's what's being shown up here. And then primary bronchial buds will form from there. Then the left and the right primary bronchi form, followed by the secondary and then tertiary bronchi and the bronchioles. So it sort of follows that same branching pattern we already learned about. The terminal bronchioles will divide into the respiratory bronchioles, which will divide into alveolar ducts. And so all of this is happening over time. So we have the estimates of when this is happening over here. Then the terminal sacs will be in close contact with capillaries around 24 weeks to term. And then after birth, those alveoli will continue to mature and develop. And then this is the last slide, some errors in development with lung embryology. So we can have tracheoesophageal defects. And so I have 4 different examples here, but there are really a whole lot of different patterns that can happen. And what it is just incorrect partitioning of the esophagus and the trachea. So the trachea is being shown in yellow here, and the esophagus is being shown in blue. And so a fistula is a connection just between things where there shouldn't be a connection. So normal development would have the trachea and the lungs entirely separate from the esophagus, because the trachea is where we're breathing, and then the esophagus is how we swallow food down to get into our stomach. So those 2 things shouldn't normally be connected. But when this is separating in development, there can be errors in that. And so this is just showing different things that can happen, different ways this can go wrong, where these connections are incorrect. And of course, that can cause problems with breathing and with swallowing and being able to get nutrition. And then one other thing that can happen if a baby is born very prematurely, they can have trouble breathing. And so that's because if they're born early enough, it might be because they don't have enough surfactant. And surfactant is really important because it keeps the alveoli from collapsing when you breathe out. And so the surfactant sort of keeps those alveoli open, makes it so they don't collapse. If they do collapse, then that will lead to troubles with breathing. And so this can lead to respiratory distress syndrome. And babies born before 28 weeks will often have trouble breathing on their own. So this production of surfactant is something that's really important for babies to be able to breathe once they're born. And I think that was the end. Yep. Okay. Any questions? Do I stop sharing my screen now?
If you want to keep sharing, thank you, Dr. Kruger. That was so great. And again, a lot to cover in 30 minutes as we always do. So if you want to just leave it shared for now, and then if anyone has some questions, then you can always skip back over to an image, and we welcome anything in the chat, or anyone feel free to raise your hands and ask away. It's probably because all of our tutors have done such a great job, and
I don't know about that. I think I went pretty fast. I almost ran out of time.
Or any questions about Dr. Kruger's journey in anthropology, her favorite system is the skeletal system, anything osteology.
Bones are my favorite, especially the skull, especially the sphenoid.
Very, very cool. All right. Hey, Michael, if you want to go ahead and unmute yourself and ask.
Yeah, I had a question. How exactly does the surfactant keep the lung open? Like, how does that work? Is it just like providing some sort of substance so it can't close completely? And what does collapsing mean as far as an alveoli, like, or alveolus? What would that mean?
So the alveoli are like little sacs, and so the surfactant is like a fluid that creates surface tension to sort of keep those sacs held open so that they don't collapse when you breathe out. Because otherwise, you can imagine a sac, if air is leaving the sac, then they can collapse on themselves and not be able to open properly again.
And
there might be more details.
Yeah, if I might, I'll just add really quickly. And it's a fluid, Michael, that has both a protein component, a water component, and most importantly, a lipid component. And so it really provides a kind of slick surface at the interface between the warm, moist air and that lipid material. And that really is what is helping to distribute the resistance. When the air is leaving, it redistributes it and helps maintain the opening of each alveolar sac.
Thank you, Dr. Peters.
Great question and great answer. Marie, do you want to unmute yourself and ask your question?
Me?
Yep.
Yeah, so in the learning objective, it says identify the innervation of the lungs and diaphragm. Did we do that?
So the innervation of the diaphragm is the phrenic nerve. So it's the phrenic nerve that sort of keeps us breathing. So that's really, oops, there it is. It's that slide. Oh, it wasn't that slide. I'm sorry. I went too far. We also have parasympathetic and sympathetic innervation that will help to constrict and dilate the bronchi. And then also some sensory innervation from the vagus nerve and thoracic ganglia going to the trachea and the bronchi.
All right, thanks.
Mm-hmm.
All righty. Any other questions for Dr. Carragher? Great. Well, thank you so much. That was so great, and really appreciate all that you did to cover all that information.
Yeah, thank you for the opportunity, and I appreciate you all being willing to listen to me for 30 minutes.
No, it's all of our pleasure. All right. So I'll go ahead and introduce our next speaker and get Dr. Elizabeth Spudik the floor. So she's joining us again this week, and I'm really happy to introduce her for this session for anyone that may have missed last week's session. Dr. Spudik is the Anatomy Thread Director at the Sidney Kimmel Medical College. That's at Thomas Jefferson University. She earned her Bachelor's of Science in Biology from Chestnut Hill College, and her PhD is in Experimental Teratology and Developmental Biology from Thomas Jefferson University. Dr. Spudik's favorite part of anatomy is whatever she's teaching at that moment. So for today, it's going to be all about the gastrointestinal system, and that's one of my favorites too. When not teaching, Dr. Spudik spends her time with her husband, Sean, and her beagles, Curly Fries and Moody Blue. Still love those dog names, so cute. She likes to knit and crochet, which, as Dr. Carragher just taught us, is a really good way to activate your parasympathetic nervous system and decrease that heart rate. And Dr. Spudik also likes to watch crime shows on TV, which is a good way to activate your sympathetic nervous system and increase your heart rate. So without further ado, I will turn the floor over and welcome another amazing presentation for the next 30 minutes.
Thank you so much. Can I share my screen? Yes, I can. Let me share that. Can everyone see my screen and my giant fluorescent green pointer? Okay. So this is the gastrointestinal system. I was here last week, and I did a whole bunch of things last week, and this is a little bit more focused today. These are the review objectives that we were given. So I'm going to go through them, not necessarily in this order, but you will know where I'm focused because I'll tell you. So we're going to start at the beginning of the GI tract with the structures involved in swallowing. So if we think about swallowing, of course, we chew and masticate and move food around our oral cavity, and then we have to move it into the gastrointestinal tract. And to do that, we have to pass it through a region that is situated behind the oral cavity, which is called the pharynx. So there's a nasopharynx, there's an oropharynx, and there's a laryngopharynx, which is aligned with our voice box. The pharynx is really a common pathway for both air and food or drink. It is guarded partly by two structures. One is the uvula, which hangs down from the soft palate. During swallowing, this is going to be pulled upwards to block off the nasal cavity so that we don't shoot milk out of our nose, although some people can on command. And then as the food is moved down into the esophagus, the epiglottis is a laryngeal cartilage that's going to close and protect our airway, okay? So that we don't choke on our food. The pharyngeal apparatus itself is really made up of a number of muscles. Now, I don't want you guys to be memorizing these very long Latin-based names, but I do want you to understand that there's a series of up and down longitudinal muscles, and then there's a series of circular constrictor muscles. And during the process of swallowing, if we focus down here on the text at the bottom, okay, when we swallow, the tongue is going to move the contents in the oral cavity to the back of that oral cavity. So that's a bolus of food or our liquids. The long muscles are going to pull the pharynx up over the contents of the oral cavity, and then the constrictors will sequentially constrict to move the contents down into the esophagus. And that is how we are swallowing. Okay, so I want everyone to take a deep breath and then swallow some spit. And then think about what these muscles are doing as you are swallowing. Maybe you're eating, I don't know, if you're eating food. I hope you know, whatever you're swallowing, think about what those muscles are doing. Okay, so those are the muscles and structures involved in swallowing. Okay, now once we have moved everything into our GI tract, we need to be thinking about control. So there is a very complex neural control of the gastrointestinal system. The gut tube as an evolutionary organ held our first brain, okay? And that was a series of neurons that now in humans make up what we call the enteric nervous system. This is the brain of the gut. You probably heard about these structures in earlier sessions. We have a group of nerves called the submucosal plexus, and a plexus is just like a braiding of structures. So we have a braid of nerves in the submucosa. We have a braid of nerves in the in between the two muscle layers, and these make up our enteric nervous system. The submucosal plexus is really positioned near the lumen of the gut and is really controlling neural control of secretions into the lumen. The myenteric is controlling the muscles, how they contract to move things along the gut tube in peristalsis, okay? So the enteric nervous system is our baseline control of peristalsis and secretions. And like we just learned with the heart and lungs, we can come in and override that baseline control with our autonomic nervous system. Our vagus nerve is parasympathetic. It's running rest and digest. And this is actually going to increase the activity of this enteric nervous system in response to substances that are in the gut tube. And so that's going to kind of activate our digestion. And then we have our sympathetic, which is our fight or flight. This information is coming from what we call splanchnic or visceral nerves. These are associated with the sympathetic chain and the sympathetic ganglia, and these are going to decrease the activity of the enteric nervous system. When we're running from the saber-toothed tiger, we do not want to be digesting any food, okay? We want to stop that down, focus all of our attention on saving our lives. And don't forget, there's always a sensory component. So if we look at this schematic here on the side, we have the epithelium in our gut tube sensing something is in the gut tube, sending that down to our sensory neurons. Some of that information is going to go up to the brain so that we can control with either the splanchnic sympathetic nerves or the vagus parasympathetic nerves. And some of that sensory information is going to go directly to our enteric system, the submucosal plexus and the myenteric plexus, to either start secretions or start peristalsis at the same time. So that is our neural control of the gut tube. The last thing with respect to the anatomy is I want to talk about the peritoneum and the peritoneal cavity. I was so glad that we just got done that lecture with all the pleura and all the pericardium, because the peritoneum is basically the same sort of structure. When we think about the peritoneal membrane, it is a serous epithelial membrane. It's going to line the abdominal cavity, okay, just like the pericardium lined the pericardial cavity and the pleura lined the pleural cavity. So the peritoneum lines and covers the peritoneal cavity. It has two components that are continuous with one another. One is the parietal component that is around the walls, covers the walls, and then there's a visceral component that invests and wraps around the organ. In between, there is a potential space, okay? It is probably a little bit larger of a space than what we would see in the pleural space or the pericardial space, because there's a lot of stuff a lot of organs in the abdominal cavity, and they have to kind of somehow fit into the abdominal cavity. But one thing to remember, there are no organs in the peritoneal cavity. The organs are covered by the peritoneum, but do not exist within the cavity. You should never have anything in the peritoneal cavity. Now, with the visceral peritoneum, because there's so much stuff that this visceral peritoneum is covering, some of the organs may be completely covered with peritoneum, and some may only be partially covered with peritoneum. Structures that are completely wrapped in this serous membrane are termed intraperitoneal. And they are things like our stomach is completely intraperitoneal, the liver, the spleen, jejunum, ileum, the transverse colon, and the sigmoid colon are all considered to be intraperitoneal. They are completely wrapped in the visceral peritoneum. The structures that have peritoneum only on one side of them, okay, these are considered to be retroperitoneal. And they include things like the pancreas, the duodenum, and the ascending and descending colons. So here we can see a transverse section. We have our peritoneal cavity, and we have our visceral peritoneum around our small intestine, and our descending colon and ascending colon are only partially invested, only like the ventral surface, okay? And here in this sagittal view, we can also see that the pancreas is not fully invested with the peritoneum, nor is this little component of the duodenum. But almost everything else is completely wrapped in the visceral peritoneum. Any questions so far? Here's my chat box. No questions in my chat box. Michael, you've raised your hand. Do you have a question?
I do. I was curious as far as like, I know the kidneys are retroperitoneal.
Yes.
But does that mean that they're also covered by that visceral peritoneum on one side, or are they something special?
No, they do. They do have a face that is aligned with the visceral, with the peritoneum. They are behind the peritoneum, just like the pancreas is behind the peritoneum. But the kidneys also have a couple of extra wrappings around them. They have a renal fascia, and then some renal fats, and then some more renal fats. And you'll hear a little bit about the special packaging that the kidneys get in, I believe, the next talk. Okay, so they are behind the traditional peritoneum, but they have a couple extra things, a couple extra wrappings going on. That makes sense?
Would they still be considered a retroperitoneal
Yes.
organ? Okay.
Absolutely.
Okay.
Absolutely.
Thank you.
Absolutely. Okay. So now we've gone through some anatomy. Let's move on to our embryology here. So I took this image from your earlier review from I guess about a month or so ago. Remember, the GI system is from the long gut tube. We have our esophagus, it moves into our stomach, our duodenum, our small intestines, first jejunum, then ileum, then out to our cecum, up through our ascending colon, transverse colon, descending colon, sigmoid colon, rectum. Okay, so we have a lot there. And that large gut tube has 3 divisions during embryology. So we have a foregut, a midgut, and a hindgut. The foregut is our esophagus down to our pancreas, where our pancreatic duct empties into, and everything associated with that. Our midgut is just past that pancreatic duct, so it's the distal duodenum, the rest of the small intestines, and about 2/3 of the way across the transverse colon, and the hindgut is the rest. So we have the distal transverse colon, descending sigmoid, and then the rectum. Now, one of the cool things about GI embryology is that each of these divisions of the abdominal gut tube has its own artery, okay? So first we have the artery of the foregut is the celiac trunk. So it's a large but short stubby vessel that comes off the anterior surface of the aorta. And from that vessel, we have all the vessels that sprout from the celiac trunk are derivatives of the foregut. So we have our left gastric artery supplying our stomach and our distal esophagus. We have our splenic artery, which is supplying our pancreas and some of our stomach as well. We have our common hepatic, which is going to give rise to our liver, supply our liver, still more of our stomach, portions of our pancreas, and our proximal duodenum, okay? So we have all of that blood supply from the celiac trunk or the artery of the foregut. And yes, the gallbladder is a product of the foregut, and I kind of always kind of put that in with the liver. So whenever I'm talking about the liver, I'm also talking about the gallbladder too. I tend to forget the gallbladder. I don't have one anymore. It got taken out, so it's kind of not on my radar all the time. The superior mesenteric artery is the artery of the midgut. So this is a long vessel, again, coming off the anterior side of the aorta, and its branches are going to supply all of the adult derivatives of the midgut that are found in the abdomen. So some of the first branches are the pancreaticoduodenal branches, and as their name implies, will supply the pancreas and the duodenum. We have the middle colic, which is going to supply the transverse colon. Off the left side, we have lots of small vessels, smaller vessels that are jejunal and ileal vessels, supplying the small intestines. And then off the right side, we have the right colic that will supply the cecum, the appendix, and the ascending colon. The inferior mesenteric artery comes off again. It's another long vessel off the anterior side of the aorta, about 2 inches above the bifurcation of the aorta, where the aorta ends and becomes the common iliacs. This will send branches to supply all of the hindgut derivatives. So we have a left colic branch that supplies the transverse colon and the descending colon. We have a number of sigmoidal branches supplying the sigmoid colon. And then the last branch is the superior rectal, supplying the rectal area. Now, another cool thing we learned about anastomoses in the last session, the last presentation, and so there is a natural anastomosis between the superior mesenteric artery, specifically the middle colic branches, and the inferior mesenteric artery, specifically the left colic branches, that allow for collateral circulation should any of the branches of these 2 major vessels be blocked. Okay, so we don't want to block the blood to our intestine, because that would be a really dire situation. Another part of embryology that I know that you guys had talked about has to do with innervation. So when we think about innervation of the gut, we are really looking at, we mentioned before, the enteric nervous system and the autonomics, both parasympathetic and sympathetic. These, as you remember from earlier sessions, the autonomic nervous system uses a kind of midway ganglion. At some point, we have a nerve from the central nervous system, synapsing on a ganglion, and then from the ganglion to the target organ and tissue. Now, when we think about that autonomic ganglion, okay, be it sympathetic or parasympathetic, one of the things you have to realize is that all of those ganglionic neurons are derived from the neural crest cell. So when the neural tube was closing, the cells at the very top of that neural tube peel away as neural crest cells. And you've heard all through these sessions how the neural crest cells contribute to all sorts of different structures in the human body. The neural crest cells are going to collect as the sympathetic ganglia, okay, of the sympathetic chain. Some of them are going to collect as pre-aortic ganglia. We'll see them in a minute. And these pre-aortic ganglia are going to control the sympathetic innervation to the entire gut tube. And those neural crest cells are also going to migrate into the gut tube and give rise to the two subdivisions of the enteric nervous system that we just talked about, the submucosal plexus and the myenteric plexus. So everything's really all kind of coming together here. We talked about the blood supply and how each of the major vessels off the front of the aorta was an embryonic vessel to part of the embryonic gut tube. Well, the same goes with the ganglia. So when I talk about the pre-aortic ganglia that is controlling the gut tube, these ganglionic cells are going to come and situate around the celiac trunk, the superior mesenteric artery, and the inferior mesenteric artery, and they will give rise to similarly named parasympathetic ganglia. So we have the celiac ganglia up here around the celiac trunk, the superior mesenteric ganglia around the superior mesenteric artery, and the inferior mesenteric ganglia around the inferior mesenteric artery, okay? So here we line up our blood supply with our sympathetic autonomic innervation. The rest of the parasympathetic innervation is really that first is from the vagus nerve, and then the ganglia again are the enteric cells in the organ of the gut tube. Okay, so let's do some histology now of the gut tube. We have our, again, from our previous, we have our gut tube has 4 layers. We have a mucosal layer, a submucosal layer, a muscularis externa, and then a serosa, which is an adventitia that is wrapped in that visceral peritoneum. But as we move along the gut tube, each region is going to tweak that layer to facilitate the function. So let's take a look at some of that tweaking. In the esophagus, one of the major tweaks is the mucosa, is a stratified squamous epithelium where we have multiple cell layers that protect that tissue, protect that organ against abrasion. Remember, we're swallowing things, hot coffee, spicy foods, and we don't want to damage the tissue too much. So we have a stratified squamous epithelium. We also see some tweaking in the muscularis layer. The upper third of the esophagus has striated muscle, and this helps us to facilitate swallowing, but it also helps to facilitate vomiting. While the lower third is just the smooth muscle and completely under the control of the autonomic nervous system. We have an outer longitudinal muscle and an inner circular muscle, and then that myenteric plexus is going to be right in the middle. In the small intestines, the job here is to absorb. So we maximize our surface area available for absorption. The first thing we do is we we send folds of submucosa into the lumen. These are called circular folds, or if you rather the Latin, the plicae circularis. Each of these folds has a number of mucosa that has been folded in and out. These are intestinal villi and intestinal crypts. So they come out and they go in and they go out and they go in and they go out and they go in. Within this villi, we have some epithelial cells that are have lots of microvilli for absorption. Some epithelial cells are goblet cells for mucus secretion that lubricate and protect. And some deep in the crypts are for bacterial immunity, okay? And these are called Paneth cells. So if we take a look at what these look like here in this upper left panel is a nice histological section. We see our plicae circularis. We see our villi coming off each of those plicae. We move to this panel here in the upper right. This is a zoomed in on that villi. We can see all of our enterocytes that have our microvilli brush border. And here's a nice goblet cell releasing its mucus into the lumen. And in this bottom panel, we can see at the base of an intestinal crypt, these cells, these are Paneth cells, and they're filled with what look like red gumballs. And each of these gumballs is holding a bunch of antimicrobial substances to keep the bacteria out of your gut, the bad bacteria. In the colon, we have no circular folds, no villi, no Paneth cells. Our muscular layer, we have our two muscular layers, but our mucosal layer is really just set up for water absorption and mucus secretion for lubrication. So here we can see lots of colonic crypts here. We can see mucus cells here, goblet cells secreting mucus, and all the rest are there to absorb water from the stool. The appendix is, we all know, is a small diverticulum that kind of a worm-like appendage that hangs off the cecum. It's kind of a little bit, half of it looks like colon, half of it looks like small intestine. The mucosa is simple columnar cells, a lot of mucus secreting cells there as well. We have our two muscle layers on the outside, but the hallmark of the appendix is the submucosa is riddled with these large lymphoid nodules. And it is believed now that these lymphoid nodules help set up the adaptive immunity for the entire gut tube. In the stomach, we have a number of adaptations as well. The muscularis external layer actually has 3 layers of smooth muscle. There's a deep third layer that is an oblique layer. And the 3 layers make sure that the stomach can move in all 3 planes and we can adequately mix our food. The myenteric plexus is still located between the first and second layer, just like it is in all the other regions of the gut tube. The mucosa of the stomach is folded into gastric pits, and then they expand out into kind of tubular gastric glands. The cells lining the lumen of the stomach and the neck of this pit are all mucus secreting cells. They're all protective. Remember, we have a lot of acid in the stomach. We want to protect those tissues. So that mucus is there to neutralize the acid and the gastric enzymes. That acid and gastric enzymes are really coming from the gastric glands, which have 4 different cell types, certainly mucus for protection. The parietal cells are the acid secreting cells in the gastric gland. The chief cells down here at the bottom of the gastric gland are the enzyme secreting cells. So these are going to secrete pepsinogen. And then sprinkled along the gastric gland are enteroendocrine cells, and these provide a local hormonal control. They secrete things like gastrin, ghrelin, and motilin to help prime the rest of the GI tract when the stomach is full. Okay, and our last set of histology, looking at the accessory organs of digestion. So we're going to start with the liver. Liver cells are called hepatocytes, and they are ranged in cords. So these are like long columns of cells. Each of these cords are fit into a six-sided plate, okay? And then the plates of cords are stacked around and aligned with the blood vessels in the liver. And each of these, and a cluster of these stacked plates is called a hepatic lobule. That lobule has a central vein, okay? And the outside, the points of that hexagon, okay? Each point has a little bit of the portal triad, which we now know, hopefully from earlier sessions, to be a portion of the hepatic artery, a portion of the portal vein, and a portion of the bile duct, okay? So here is a schematic where we're looking at just kind of one corner of our hepatic lobule. We can see that the blood is moving from the portal vein and the hepatic arteries, moving towards the central vein. The blood is filling these hepatic sinusoids. It's percolating through all of the hepatocytes. And as the blood is percolating through the hepatocytes, they are detoxifying. They're taking the nutrients out and metabolizing them, the sugars. They are metabolizing the hemoglobin that's being dumped from the spleen into the blood, and certainly getting oxygen from the hepatic artery. As they're doing all this, they're also making bile, and bile is moving away from the central vein into the bile duct. So the blood is moving in one direction, and the products of the hepatocytes, the bile, are moving towards the opposite direction. The gallbladder, I didn't completely forget the gallbladder. The gallbladder is really adapted for storage, concentration, and controlled release of the bile that is made by the liver. So we have a mucosa that is a simple columnar epithelium, and it's really set up to concentrate and absorb fluids from the bile. The muscularis externa is very, the muscles are arranged haphazardly, again, so that the bladder can contract in all directions so that it can effectively push out the bile when needed. And then we have the pancreas. The pancreas is two glands in one. We have an endocrine gland function where we are secreting hormones directly into the bloodstream. This is from the islet cells. And the two major hormones that are being secreted there are insulin and glucagon. And then we have an exocrine function that is secreting digestive enzymes. And these are going directly into the duodenum. And these are being secreted by these glandular acini out here on the periphery. And we make about 1.5 to 3 liters of pancreatic fluid per day. So that's a lot of fluid being pumped by that pancreas. And here we have a histological section. We can see our islet cells right here. These are the endocrine portion pumping out insulin and glucagon. We can see our little rosettes of secretory acini here around the islet cells. And here's a little part of the ductal system that is going to be taking the secretions of this acini and moving it into the gut in the duodenum. Okay. And that is it. And if anybody wants to see, these are my two puppies. But I will take questions. What are the questions already in the chat? Okay. The non-oblique, so there's always a longitudinal and there's always a circular in the gut tube. So there's a longitudinal layer, then there's a circular layer, and in the stomach, we add an oblique layer, okay? So that we have one going up and down, one going around, and then a third at different angles, okay? So that we can get not just a contraction, not just a shortening, but like a twisting as well when we move those muscles. Do the hormones released by the enteroendocrine cells in the stomach help activate? So those hormones are really, they're activating the enteric nervous system. That's probably their primary target, to prime like the very next region of the gut tube, but they will circulate as well and have effects on the central nervous system. That signaling is very, very complex. I don't want you to get too bogged down on it. But I want you to know that those cells, when the stomach is full, they are saying, hey, we need to prep for what's coming down the pipe. So they're sending out very local hormonal signals to the next section of the GI tube. Let me just see here.
Looks like we've got a question from William. I just wanted to say thank you for that presentation. That was very thorough and again, very condensed for what you
Very
taught.
condensed.
Very good.
I
Very
could
good
talk all
representation.
day.
Yeah, I know. Right?
Who wants to unmute themselves first?
I think William had his hand up first. We'll let him go
Yeah,
first.
so did we ever say what the purpose of bile was?
Oh, so bile is to emulsify fats and lipids and cholesterol.
Does it
So
is that what emulsify means?
yeah, so it's so remember the lumen is largely a water-based, so the gut tube inside is largely a water-based system. And in order for the fats to be effectively metabolized, they have to be because they'll separate out from the water, just like a salad dressing. Okay, the bile helps to kind of put a charge onto the lipids and allows them to remain in solution so that they can be taken up by the cells of the gut tube and moved into the circulation. Does that make sense?
Yes.
Who else?
That would be me. That was a good question.
Who is me?
Michael.
Hello, Michael.
So the you mentioned the liver picked up hemoglobin from the blood. And you said like this, the spleen deposited it. So does that mean the spleen destroys or like dismantles the worn out red blood cells and then releases the wastes, and then the liver picks up the wastes and metabolizes
Absolutely.
them?
You're
Okay,
right.
cool.
You're bang on. So red blood cells, the ones that are tired and have been used a lot, they are broken down by the spleen. They are dismantled. They're chopped up by macrophages, and the reusable parts, like the iron and the hemoglobin, are sent back to the liver, and the liver is going to process them. And that's a whole big, it's going to be a whole big set of biochemical reactions at some point that you guys are going to love to learn. But you're absolutely right. The spleen breaks down, sends the iron and stuff back to the liver to be processed and reused. And then, and part of that, what can't be reused is moved into the bile.
Well, actually, how does it know which red blood cells are like worn out and like ready to be recycled, and which are like still strong?
So as the cells are moving through your body, they are picking up other proteins on their surface. They are sometimes getting a little jagged. And the spleen, the macrophages in the spleen will see the other proteins that are stuck onto the surface of the red blood cells and pull them out. So that's a very simplified version of how we get rid of red blood cells.
Would that larger amount of proteins also slow them down when they're trying to go through the spleen?
No, it's it doesn't tend to slow them down. It may make them a little bit, like I said, more likely to be recognized by a macrophage as kind of a, you know, a little tired.
Okay, thank you.
Yeah. I think we could go on all day. My email's at the front of this. Email me and we'll talk all day about red blood cell clearance.
All right, looks like we got another question from Riva. Do you want to go ahead and unmute yourself?
Yes, thank you. So you might have already mentioned this, but I was wondering what part of the red blood cell can be salvaged?
So there's actually so the red blood cells are going to be taken up by the macrophages, okay? And most of what is there is recyclable in some fashion, but it's chopped up and then it's going to be kind of used and kind of like broken down, almost like a garbage disposal. Okay. And then a lot of that stuff will go back kind of into the fluid of the blood, and it will be like the individual amino acids will be reused, the individual components of hemoglobin will be reused. So it'll circulate back into the bone marrow where new red blood cells are being made. So it's a very, very complex process. But just like every cell in your body that is used up and removed through apoptosis or just an old cell is going to be kind of taken in, chopped up, and then all of its component parts, all of its proteins, amino acids, nucleic acids will eventually be recycled. Our body doesn't waste a whole lot. It would be inefficient.
Excellent. Thank you so much. I think we're going to go ahead and move on for our next speaker to make sure we get through those. If you guys still have questions, feel free to keep putting them in the chat and we can try to still address those for you. But I'll turn things over to Dr. Peterson to introduce our next speaker. And thank you again, Dr. Sputic, for a lovely presentation. Very informative.
Great.
So hello, everyone. It's my honor and my pleasure to introduce Dr. Catherine Veazey. Dr. Veazey it has earned a master's degree from Colorado State University. Her master's degree is in biomedical sciences, and then she relocated to the fabulous state of Mississippi, where she earned her PhD in clinical anatomy at the University of Mississippi Medical Center. She is our newest faculty member in the anatomy department at Drexel University. We are thrilled to have her. Her research interests currently include clinical communication skills, both with medical students in the hospitals, but also with interprofessional healthcare teams. And the fun fact about Dr. Veazey, just pet trivia tonight, she has a dog named Maisie, and she often describes it as looking like a cow. So there you are. So Dr. Veazey, please feel free to get started.
Great, thank you so much. Can you all see the screen? You see the pointer? Okay, wonderful. So I'm here today to talk about urinary and reproductive system, which are two of my favorite things to talk about. A lot of objectives to cover. We won't necessarily cover them in order, but we will mainly be talking about the innervation of the kidneys and bladder. So I actually do not have any slides on the gross anatomy or the relationships of the fascia, but if we have some time at the end, I'd be happy to talk about those during the question segment. Otherwise, we'll be focusing mostly on the histology and the development of those structures, as well as the female and male reproductive systems. I was set up very well by the previous two speakers because now you should all have a pretty good understanding of some of your body cavities. So during development, we are going to segment everything into a series of cavities. We've got our thoracic cavity superiorly, which, as we now know, contains the mediastinum, which has multiple parts at the center, as well as two pleural cavities on either side. We then have our peritoneal cavity that will be inferiorly, which will be mostly associated with our abdominal cavity and continue partially into the pelvic cavity. From an embryological standpoint, we really break these into the dorsal cavities and the ventral cavities. So all the ones I just named are ventral. Ventral is also towards the stomach, so in this case also anterior. We then also will have our dorsal body cavities, including the cranial cavity, which will consist of our brain and our brain stem, and continue down into our vertebral cavity, which will contain our spinal cord. So each of these cavities have their own viscera or organ systems that are then associated with their own linings. So as you now know, the lungs will have the pleura, the heart will have the pericardium, and the abdominopelvic structures will have the peritoneum. So we are going to actually form and segment these structures in a stepwise fashion. So during the developmental process, I was told you've already had a little bit of this before, so I'm starting somewhere around week 3 and 4, where we've got body folding happening. So key things to keep in mind with the process of body folding, we're going to have what we call craniocaudal folding, which is when the head and tail start to fold in towards each other, as well as lateral body folding, where we've actually got the sides of, at this point, what was our trilaminar embryonic disc, which just means it had 3 layers, now folding to actually form what we call the tube within a tube model. So it looks like a fun multicolored donut. So this creates a bunch of things, including our intraembryonic coelom. You're going to see this word a lot. It's pronounced a lot of ways. I call it the coelom. You might see it called the chelum or the coelom. But all that this word means is a body cavity. So this is just the primitive body cavity that we find when we actually fold up this lateral wall to pinch together. And remember, we have this funky little thing called the umbilical vesicle. You may have seen it referred to as the yolk sac, which is actually sticking out the front of our little embryo that will be associated with the development of some of our gut tube structures that you just saw in the previous talk. So as this all is happening, we're going to start forming our little heart tube, which will form with its associated cavity, the pericardial cavity. We can see this on midline, but it will also be forming its pleural cavities on either side. And then inferiorly, we'll be building our peritoneal cavity. So we can look at all of this in a bit more detail on the next slide. So keeping in mind, we're looking at a cross-sectional view here. So the back of the embryo or dorsal is towards the top of the screen, ventral or anterior towards the stomach is at the bottom of the screen. And so we can see we've got our little spinal cord developing at the back. We've got our heart tube developing with its aorta, and we've got our lovely little trachea and esophagus developing on midline. We can then see we've got the heart suspended now anteriorly in this little primitive body cavity. So again, this will become the pericardial cavity. And now we start getting into the incredibly long words. So you're going to see these words and they all are abbreviated PP something. So just break it down by the cavities we're trying to connect. So along midline, I'm trying to connect my heart cavity, which is the pericardial cavity, down to the developing abdominal cavity. So I have the pericardio-peritoneal canals. Lateral to that, I will also have the pleuroperitoneal canals, connecting my developing pleural cavity, which is where the lungs will go, down to the sides of the abdominal cavity. As this is happening, we're going to actually anchor our developing gut tube, represented here by this little sign for stomach and the liver. Remember, these are structures of the foregut. We are anchoring them to the body wall by what we call mesenteries. So we have a ventral mesentery, which suspends almost all of it, as well as a dorsal mesentery, which connects it to that posterior body wall. Remember, dorsal just means back, ventral just means stomach. So this dorsal mesentery is actually going to form most of the structures that were talked about last time that are actually anchoring these. So intraperitoneal structures will develop within and stay within these ventral and dorsal mesenteries, while things that are retroperitoneal either developed completely separate, as we'll see with the kidney, or developed within these little mesenteries and then lose their connection along their dorsal surface. So I know the pictures start to get really complicated, but keep in mind all of our orientations are the same. So along the back, we've got dorsal with our little developing spinal cord, and we've got ventral with our developing heart. So we can see this heart cavity, and we can see this pericardio-peritoneal canal. We're going to build a series of folds. Firstly, our pleuropericardial fold, which is going to divide our pleura, or the two lungs on the sides, from our developing heart at the pericardial cavity. So we can see these come in and form this nice little membrane that will separate our heart and our lungs completely. And initially, this is really separating them from anterior to posterior, but as the lungs start to get bigger and occupy more space, they will begin to kind of curl around so that they are really located both lateral and a little bit anterior to that heart cavity. As this is all happening, we then have our second set of folds, the pleuroperitoneal folds. The image unfortunately doesn't really show the folds so much, but it does show this lovely little canal where we've got that little developing lung separated now from the structures of the foregut. We've got the liver over here, and it doesn't label it, but the spleen would be on the other side. And so we can see how that would be separating it along the lateral segments. As all this is happening, we have a new structure, septum transversum. That just means a septum, which is something that divides two things, that is transversely oriented. So it's just a pretty much flat line dividing our embryo along this transverse plane. Looking at this septum transversum a little bit more, this becomes most of that diaphragm that was talked about in the very first session. So the diaphragm is actually going to form from 4 different tissues. Again, the septum transversum forms most of it, but we're also going to have some contributions from this mesentery of the foregut, specifically at the region of the esophagus. This will form several openings for structures to pass between the thoracic and abdominal cavities, including openings for the inferior vena cava, the esophagus, and the aorta. We will then have contributions from those pleuroperitoneal folds. We can see these in hot pink, magenta, pretty brightly saturated color, which is forming this posterior lateral segment. And last but not least, there's a little bit of musculature that's actually growing forwards from the body wall to form the rest of that diaphragm as it tenses over those cavities. So that's the overview of where we put things. Now we can talk about the systems themselves. So first off, urinary system. I'm told you've had some introduction to the system. So mainly what you need to know, we've got a kidney, a ureter, the bladder, and the urethra. And the whole purpose is to make and release urine. The kidney is what we're focusing on today. It has a lot of parts, including the cortex, which is the outer section, as well as the medulla, which is the inner section. This is segmented into a series of pyramids, which will taper down to form these little papillae. The papillae will drain into our excretory system. So these are going to be called calyces. We have minor calyces that drain into major calyces, then through the renal pelvis and out through the ureter. They're supplied by a series of renal arteries and veins that will form this very dense vascular network along the medulla and the cortex. The key functional unit of the kidney is the uriniferous tubule, which really has 2 parts. We've got a nephron, which has lots of subparts indicated in red, and we have the collecting ducts. The collecting duct is part of that excretory system. It's going to get us out of that renal papilla and into those calyces. So looking from left to right, we're going to have this little glomerulus, which is actually a set of capillaries that is associated with this capsule. So the capsule is going to be the actual first part of our filtration system of the kidney. Those two together are called the renal corpuscle. You may also see that called Bowman's corpuscle, but we'll be calling it renal. From there, we have the proximal tubules. These are both convoluted, which just means very twisty, as well as straight. We then enter our loop of the nephron, most often referred to as the loop of Henle. From there, we get into our distal tubules, both straight and convoluted, and then out through that collecting duct. So looking at each of these in turn, we can start in the renal cortex. So we're going to first have the renal corpuscle. The corpuscle looks like a ball of string. That's what corpuscle means. So anytime you see this really large mass of cells and dense tissues with this big gap around it, that's what you're looking at. So the glomerulus is inside of it, and this little capsule on the outside would be that renal capsule or Bowman's capsule. From there, we have the proximal tubules. Proximal tubules, whether convoluted or straight, are very acidophilic. That just means they're incredibly pink. So they really are very densely colored. and they have a pretty significant brush border that makes their lumen, or that hole where they drain all their fluids, very hard to see. So when you can see it, it looks stellate or star-shaped, but often it can be completely occluded. So in this example, again, you can see in the illustration, we've got these hot pink cells with pretty clear nuclei and a very fuzzy lumen. So it looks like it's full of stuff. We can contrast this with the distal tubule. Distal tubules are more pale. They actually have a fairly distinct lumen, and oftentimes the nucleus will actually bulge into that lumen. So in this example, you can really see these cells starting to bulge in to that much larger luminal space. Last but not least, we've got these collecting ducts. A collecting duct can look a lot like a distal tubule, but the main thing you're looking for are these very distinct intercellular boundaries. So it looks like someone went and actually took a little pen and drew a line between each cell. So you can see these really distinct boundaries here in the indicated example. If you're lucky, you'll also see that the cells will alternate their colors. So you'll have a light cell, then a dark cell, then a light cell, then a dark cell. You should hopefully see that on the next slide. It's the slide after this, but we'll talk about it. So if we move down into the renal medulla, now we're further in that uriniferous tubule. So we've really gotten rid of all our convoluted stuff, and now we're starting to look at mostly that loop of Henle. So we've still got collecting ducts. We find those everywhere. So still indicated by this light blue line. Now, once again, you can see these really clear intercellular boundaries. Someone just drew a little line between each nucleus. But we can now start to see the loop of Henle itself. This is simple squamous. Squamous just means a flat, smushed cell. And so pretty big lumen and very little cytoplasm or cytosol. So you just see kind of a lot of little dark nuclei and a big empty space. These may be easy to confuse with the vasa recta. Vasa recta are straight blood vessels. Specifically, they're straight capillaries. Depending on where you're at, they might be peritubular capillaries. For the sake of time, we're not going to get into all of those semantics, but just understand that these are going to help us with our exchange mechanism. So we've got that big tube coming down, which is full of what will eventually become urine. We're going to put it right next to our blood vessels so we can move things in and out of them. And there will be a diagram about that in just a second. So these are also simple squamous. They're a little smaller, they're a little flatter. And if you're lucky, you'll actually see your little red blood cells occupying that luminal space. So if we contrast these directly, we can see the structures within our renal cortex. So first things first, if you ever see a renal corpuscle, don't know what happened with the cursor. If you see a renal corpuscle, you know you're in the renal cortex. Versus if you just see a bunch of straight tubes and lots of really thin, simple squamous openings, you're probably going to be in the renal medulla. These proximal tubules here in the cortex are also fairly distinctive. You shouldn't really be seeing those much by the time you've gotten down pretty far into the renal medulla. So once again, they're very pink. They've got this really distinct brush border. The lumen looks like it's just full of stuff. Versus if we compare that to our distal tubule, remember, a little more flat, big, clear, open lumen with these little nuclei bulging in. Here we can see that example I was talking about. These collecting ducts, the cells are different colors. So we've got some dark cells and we've got some light cells. We've also got those clear intercellular boundaries. So all of this is related directly to the function of these different parts. So the renal corpuscle's job is to collect all of the stuff it can from the blood. So remember that we would have our glomerular capillaries in this space. Things will move from those capillaries into the actual tube itself. So from there, as we move into the proximal tubule, we've got that brush border, all of these beautiful little microvilli to reabsorb all of, not all, but almost all of the things we put into that fluid. So you can see here, most of what's getting pulled back into the body to use for energy or for neurotransmission, all of the key functions that we need to do, it's getting pulled in immediately through that proximal tubule. From that point, we'll continue down into the loop of Henle, which would again be associated very closely with arteries on either side of it. Each part of the loop of Henle does a different thing. So some portions are permeable to water, they allow it to pass in and out, while some are not. And so instead what we get is just a series of very regulated ion transporters that let us move different solutes in and out. By the time we get into our distal tubule, we don't need microvilli anymore. Instead, we just have, again, a lot of regulated ion channels so that we can very finely tune the concentration of our urine or the filtrate at this point versus what we're actually putting back into the body. Last but not least, we get into the very end. We've left the nephron and entered the collecting duct itself. This is the final chance to concentrate or dilate the urine. So this is what is going to let you know if it's going to be very bright yellow or very clear based on your hydration level. As far as innervation goes, once again, we're going to have autonomics. So you have sympathetic activity and parasympathetic activity. The sympathetics are going to be responsible for suppressing contraction of the bladder to allow it to fill. So if you think about this, if you're running through the woods from a bear, you don't really want to pee yourself. You'd like to keep that from happening. So we're going to allow it to fill and prevent it from emptying. This is done through what are called hypogastric nerves, which just means below the stomach. So these are actually passing from the abdominal cavity down into the pelvic cavity to work on the bladder. The parasympathetic nerves are actually from the pelvic splanchnic nerves. These are from the sacral spinal cord, and they do the opposite. So they're going to tell the bladder that it is safe to empty. So again, if you're reading a book, you're relaxing, you're having a nice time, your body knows that it is safe to go and voluntarily empty the bladder. But the voluntary part is actually regulated by its own nerve, the pudendal nerve. The pudendal nerve is the nerve of the pelvis or the perineum, which is the region of the groin. And it lets you choose when to urinate. So you actually have to train your bladder via this nerve to not continually void. The kidneys will also get sympathetic and parasympathetic regulation. So sympathetics come from thoracolumbar splanchnic nerves. That's all these red ones right here. We can see them coming down, and it shows just this bottom section going to the kidney, but the literature is really all very confused about this. Just keep it general. It's thoraco and lumbar levels. Splanchnic nerves just mean nerves to organs and viscera. These are going to cause vasoconstriction of those vessels going into the kidney to decrease the rate at which we are filtering out urine. Parasympathetics are going to act through the vagus nerve, which is one of our cranial nerves. It's not very well understood what those do yet. As far as development of the kidney, it comes from intermediate mesoderm, and we actually form it in three parts. We have the pronephros, which is the first kidney. It pretty much immediately regresses and isn't going to contribute anything major. From there, we go to the mesonephros. The mesonephros is second. It's the intermediate kidney, and it forms two things: the mesonephric duct, which will contribute a lot to the reproductive systems, and then the ureteric bud. We're going to keep a pin in that for a second. Last but not least, we form the definitive kidney, the metanephros. Metanephros forms the metanephric blastema. So looking at each of these, we can now see it color-coded. The structures from the ureteric bud are all of the structures of the excretory portion. So we have our ureter, we have our calyces, and we have those collecting tubules. The nephron itself, from the glomerular capsule through to the distal tubules, is coming from the metanephric blastema. So we can see it's starting here. You'll see Wolffian duct a lot. That's the mesonephric duct. We can see this little bud, that ureteric bud, which will be surrounded by this metanephric mass or blastema, and continually grow and invaginate to form this very convoluted set of tubes, that uriniferous tubule. The capillaries of the glomerulus will come in from just the surrounding tissues. That lets us pass into the reproductive system. So I've been told you've already seen a general overview of this. This is just a big summary of all of the parts. We're really going to just be focusing on the testis and then the ovary and the uterus. But first, we have to talk about the pelvic floor. So the pelvic floor is going to be supporting all of these pelvic structures. It is going to resist intra-abdominal pressure, which allows us to maintain our continence. So again, we want to voluntarily choose when to void either the bladder or the rectum. And so this is going to allow us to do that, as well as to keep help prevent anything from moving out of this pelvic space, which is called a prolapse. It has two main components. So just to help you orient here, we are looking down into the bowl of the pelvis. Anterior is at the top of the screen and posterior is at the bottom of the screen. We can see the sacrum here. So along the anterior view, most of the pelvic floor is made of this muscle called the levator ani. Levator ani has several parts. You're not responsible for knowing them, but it's going to connect our pubic bone and our ilium back towards our coccyx. It has innervation from the sacral plexus, which is S3, S4, S5. And just behind or posterior to that, we have this coccygeus muscle. Coccygeus is a little triangular muscle attaching the ischium, which is a third of our pelvic bones, across to the rest of our coccyx. It's innervated primarily by S4. These two together create a paired muscle, which we call the pelvic diaphragm. And it is that pelvic diaphragm that will mostly help us support all of those pelvic structures. Now, we're going to spend the rest of the time on development and histology of the reproductive systems. So starting off, we're starting with spermatogenesis, which is the production of our mature spermatozoa. We do this in several stages. First, we have our spermatogonia, which are along the basal portion. So that means they are on the outside of the tube. This whole thing is called a seminiferous tubule. This is the functional tube of the testis itself, and each layer of it will contain different types of cells, at least in humans. So we start off with our spermatogonium, which again are going to be on the very outside of our little tube. They're very small, they're very round, they're also incredibly dense. We move up and we'll move into our primary spermatocytes. These are larger, they're much less dense, so they're what's called heterochromatic. I actually think that's a typo. These are euchromatic. So sorry. They're much less dense. So the big thing is their nucleus is very big. It's much lighter because what we're actually doing is unpacking that DNA so that we're able to replicate and divide it further. So you can see they're very big and very light staining. Secondary spermatocytes divide incredibly quickly. So you're not really going to see them. But a key thing happens as we move outwards towards this lumen, and it actually happens at this primary spermatocyte level. The primary spermatocyte will move from the basal compartment, which is again towards the outside of the tube, into the luminal compartment, so towards the center of the tube. These are blocked by what are called Sertoli cells, which are marked in the image as nurse cells. You may see them called that. They're not super well to see in this image, but you can see one right here. They are a little bit tall, more of a columnar cell, and they've got a big, dense nucleolus. So they've got a little bullseye in them. And they're just separating those two compartments from each other. So we're going to pass through from our primary spermatocyte. Secondary spermatocytes died immediately. And then we form our spermatids. There's actually two types, but you can just call them spermatids. So spermatids are all the cells you see lining the lumen itself. So these are now going to be pretty small and round if they're early, but they can also start to look like actual mature spermatozoa. So we can see they have become kind of small and triangular. The nucleus is very compact, and most importantly, they've developed a tail. Oogenesis is a very similar process, except it's going to have a bunch of different time points at which it has to stop. So before birth, all of the eggs are oogonium. However, by the time birth happens, all of them have completed mitosis and started meiosis 1 and then get stuck in prophase, specifically prophase 1. So by the time birth happens, all of the eggs are now primary oocytes. They will not resume meiosis until puberty happens, and then they only resume it if they're going to be ovulated. So you will only see a secondary oocyte in a follicle that is about to be ovulated. They will then freeze again, now in metaphase 2, and they will not complete meiosis until they are fertilized. So fertilization doesn't happen, you'll never actually get a mature ovum. So we're really focusing on these two because this is what you'll be able to see histologically. So each oocyte is going to be associated with a follicle. The follicle is the shell of cells around the oocyte. And as we now know, pretty much every follicle that you see is going to have a primary oocyte at its center. So we start with a primordial follicle. From the primordial follicle, we're going to have a primary follicle, which is going to add our first official layer of cells, the granulosa cells. From there, we're going to build a secondary follicle. We've got several layers of granulosa cells, and we now have a new layer on the outside called the theca, or the thecal cells. It's possible at this point that you start to build a little fluid cavity called the antrum, but the antrum will really help us define our tertiary follicle, which you might see called an antral follicle. So at that point, it's got a very big antrum, it's got lots of layers of granulosa cells, and our theca has actually divided itself into two distinct parts. We have a theca interna, which is closer to the granulosa cells, and a theca externa that is external to them. At this point, the tertiary follicle will turn into our mature follicle. That oocyte will continue meiosis and become a secondary oocyte. Remember, it will immediately stop again in metaphase 2, and it will be ovulated. When it's ovulated, we're going to lose almost all of our follicle. So we're going to let our little secondary oocyte escape out into the peritoneal cavity to be grabbed by our oviduct, or the uterine tube, or the fallopian tube. We're going to have some granulosa cells go with it, but most of it actually stays behind to become a structure called the corpus luteum. And we're actually going to look at all of this on these next few slides. So here we are in the ovary. Within the ovary, we have a cortex on the outside. We've got a medulla on the inside. We can see just below this cortex, we've got all of these little follicles, and these are all primordial follicles. We've really just got the oocyte with a pretty big nucleus and not really anything else happening. However, we can start to see one of our primary follicles now with one layer of cells around it, which would be the granulosa cells. From there, we can move to our secondary follicle. We've got a lot of layers of cells now. We've also got the start of an antrum. And then our tertiary follicle, which has all of our defining characteristics. We have the oocyte, lots and lots of granulosa cells, and now two distinct layers of theca. We have this theca interna. We can see the color changes from pretty deep purple to a little more sparse, more pale. And then we have a theca externa, which actually looks a lot like smooth muscle. The development of this oocyte is happening during something called the ovarian cycle. So we grow from our primordial to our primary to our secondary to our tertiary follicle. And as this is happening, we actually start the whole process with a lot of follicles. So several primordial follicles develop at the same time, become primary, and then they start to undergo atresia. So atresia is a controlled cell death of the oocyte and its follicles so that only one follicle is ovulated each time. These follicles can undergo atresia at any point, including just before ovulation. So the development of the follicle from primordial to tertiary is called the follicular phase. And then after the follicle has been ovulated, remember, we leave it behind as the corpus luteum. So we call this the luteal phase. This corpus luteum is defined by luteinized cells. So our granulosa cells now become the granulosa lutein cells, which are even bigger, very pale staining. They produce a lot of hormone, especially estrogen and progesterone. We also then have externally to that our theca cells, which are now theca lutein cells. These are pretty dark, pretty compact, and they produce some androgens, as well as a little bit of progesterone. The corpus luteum will stay active for somewhere around 10 days, and if fertilization doesn't happen, that is what triggers menstruation. So we can see its relationship to now our uterine cycle. So it actually starts at menstruation. So if fertilization doesn't happen, the lining of the endometrium will shed, and then the new follicle phase will begin again. So as our follicle starts to develop, the uterus endometrium will proliferate, ovulation will happen, and it will begin secreting a variety of things through a series of glands that will then, as this corpus luteum goes away and stops working, shed and shed and shed to resume menstruation. So the follicular phase lines up with menstruation and the proliferative phase. Looking at all of this real quick, because I do want to be very respectful of your time, we can see the uterus at a low and high magnification. We have the myometrium, or the muscular portion, and this endometrium. The endometrium, we can see during the menses phase, doesn't have much going on. There's not a lot of glands, there's not a lot of tissue, not a lot of vessels. It looks like it's all sloughing off, and that's because it is. Compared to our proliferative phase, we've got a much thicker endometrium. It's got a lot of these straight glands in it. They're pretty small. And notice how pretty much none of them are actually going all the way up to the lumen. In the secretory phase, these glands are now very large. We call them saw-toothed because they're very jagged or rigid around them. And several of them will actually go all the way out into the lumen. If fertilization does happen and menstruation is avoided, we're going to build a placenta. The placenta has a lot of components. Its whole job is to separate the fetus from the tissues of the uterus itself, as well as facilitate the exchange of nutrients from the maternal circulation to the fetal circulation. It produces human chorionic gonadotropin, which is actually what most pregnancy tests are going to recognize. This is what we call maternal recognition of pregnancy. And it has two parts, the fetal part, which is formed from one of our fetal membranes, this blue one right here. This is called the chorion. We also then have the parental or maternal part, which is the decidua. The decidua is just the pregnant endometrium. So the entire endometrium becomes the decidua, with the part closest to the fetus becoming a special part, the decidua basalis. If pregnancy happens, we also need to talk about the mammary glands. So the mammary glands are a modified sweat gland that during lactation will produce milk. They have several parts, including these little lactiferous ducts, which will come from our little secretory alveoli. This is the part of the gland that makes milk. They'll go out through the duct and collect in these very large dilations called sinuses, and then be ejected out through the areola of the nipple. We can see the structures here. We've got these simple cuboidal ducts. They're pretty big, lined by some connective tissue. We've got lots of these secretory alveoli, and they're associated with myoepithelial cells, which are little contractile cells to help actually eject the milk. In an inactive mammary gland, there's not much going on. It's mostly connective tissue, a little bit of fat. The glandular components are not well developed, and the ducts are basically non-existent. Versus in an active gland, we can see it's very, very different. So now we've still got fat, we've still got connective tissue, but almost the entirety of it is made up by these very dense, very active alveoli. So the cells themselves are now big, they're very purple because they're producing lots of things, including protein and lipid, and they are actually putting that product out into their ducts. So the ducts should be pretty full. Thank you so much. I know I went a little over, and I'm so sorry, but hopefully we've got some time for questions. I know the chat's been going crazy, so I'm going to open it up. Yes, Michael, go ahead.
So what's the timeline for like the so originally it's mitosis, and then you're getting all these cells that are going to become the follicles, or the oocytes rather, not the follicles. And then what for meiosis 1 and then meiosis 2, when is that happening for the oocytes? Does that happen like before they become primary follicles? Because I don't know how it would become like I'm not sure how meiosis would happen after it's already got all these extra cells around it. So it's like, when does that happen in the timeline of development?
Yes, so very good question. So the division of the oocyte is really pretty kept separate from the development of the follicle itself. So really, we've got some stuff happening before birth, which is the division of our oogonium into our primary oocytes. And then during and after puberty, so from puberty until menopause, you've really just got one division that's going to happen in the oocyte itself. And that happens right before ovulation. So we go from primary oocyte to secondary oocyte. We can uncouple that almost from the follicle development. So all of that is just associated with our primary oocyte until ovulation happens. Does that make sense?
Wait, so meiosis 2 happens right before the oocyte is released, like ovulation?
Yes, so meiosis 2 will start. So we can see in the diagram, the primary oocyte is stuck at prophase 1 in meiosis 1. Then, as we are going to develop, we'll resume meiosis 1, we'll start meiosis 2, but then we'll get stuck again at metaphase 2. So meiosis 2 is not going to be completed unless fertilization occurs. So most oocytes never actually complete meiosis.
So does that mean that it has 2 chromosomes until sperm penetrates the eggs, like the oocyte's shell?
Not
Or
necessarily. I was like, it's because at this point we're still the secondary oocyte is a haploid cell. it just hasn't completed its final division. So what we do is we actually form these little polar bodies where we put all of the extra in it. But by the time that we're secondary, we are in metaphase 2. So it's similar to what we do with our spermatogenesis. Your secondary oocyte is just like your secondary spermatocyte. They are both haploid. We just don't quite finish.
Okay, that's really cool. Thank you.
Yep. All right, William.
Yeah, hold on. Can you go back to the spermatogenesis slide?
Mhm.
I feel like, let's see, where do the actual I forgot my question, but I just missed this part. So like, what was everything? What were you talking about the where the spermatids basically?
Did you say, could you repeat the question?
I think
Could
Dr.
you like
Veazey, that William,
verbally with the spermatids where I missed it?
Okay, yeah. So spermatids. So there's early and lates. I was told you're not responsible for distinguishing between the two. You just need to be able to say it's a spermatid. But that's all of the cells that are actually lining the lumen itself. So these are, once again, these are haploid cells. They aren't quite fully mature yet. But the late spermatid, which is the most characteristic, is like a little triangle shape. So it's become really narrowed and tapered. And most importantly, it gains a tail. Which is called a flagellum, which lets it swim.
Okay, thanks.
Okay, I see a question in the chat, which is Sertoli cells. Sertoli cells are the other cell. So they are not any of the sperm cells. They're going to support the development of the sperm. So a Sertoli cell, we can see one down here. Sertoli cells are also called nurse cells. They're also called sustentacular cells. They got a bunch of names, but they're just a support cell. So they're going to look very different from the like developing sperm cells. They are long and almost columnar shaped, and they've got like a bullseye nucleolus in them. So I know that the line is covering the bullseye of it, but it just looks like a big cone and a black dot at the center.
Okay, thank you. What about the Leydig cells actually? Where are they in
Yes.
this picture?
They are actually not in the picture very well. Let me move this out of the way. So the Leydig cells are called interstitial cells. So we've got this interstitial tissue here. Interstitial is just like between adjacent seminiferous tubules. So they're not in the tube itself, they're just outside of the tube. And Leydig cells are mostly going to be producing testosterone, which is also incredibly important for spermatic development.
Okay, thank you. I had one more question was, I feel like I've asked a similar one to this, but how does the, like as it's going through the blood's going through like the nephron being like filtered, transformed into urine, how is it propelled through that? Is it from the blood pressure at the glomerulus that kind of pushes all that fluid through the nephron, or is it and collecting tubules, or is it like or is there like smooth muscle or something else that helps?
I gotcha. So yes, it's all about pressure gradients and osmolarity. So basically what we've got going on, so in the blood vessels, we're keeping the pressure very well regulated. But remember, they're going down in parallel really with our little loop of Henle. So let me go back to that picture. So we've got our part of the nephron, and then we've got all these blood vessels that are with it. So the blood vessels, again, have a very high, it's not very high, but very well regulated pressure gradient, while we have a very well regulated osmolarity gradient, which is like the amount of stuff suspended in fluid inside of the tube. And so it is actually both a matter of a pressure curve over in the vascular network, but also this kind of volume relationship in the tube itself. So it's very complicated physics and a whole bunch of regulated mechanisms that just cause things to be moving continuously in and out. And as more fluid is brought into the top of the tube, it's naturally going to be propelled through because it doesn't have anywhere else to go. So there's not typically, except in, there's a cell you don't care about, But most of this is not associated with smooth muscle, so it's not getting like squeezed to propel anything out.
Ah, okay. And throughout this entire thing, it's got like heavy heavy specialization, I assume, of like the tube material that only allows like certain things to cross over into the capillaries and
Yes,
veins.
so that's what this is trying to show, is that each part of the tube only lets certain things in and out. So it is incredibly regulated process.
Okay, awesome. Thank you.
And then I'm
Thank-
also happy to, I'll put my email in the chat in case people have follow-up questions, but thank you all so much.
Thank you. Excellent, excellent, excellent stuff. Thank you to all of our presenters for another just great tutoring session and really helping us elevate our knowledge in anatomy, histology, and embryology. So with that, that's the end of our tutoring session for today. And just want to remind you again that the next session is going to take place on March 28th at our regularly scheduled time. That is on a Thursday, I know, but hopefully as many of you guys can attend that time and date as possible. So looking forward to seeing you again there. And best of luck, everybody, in your studies, and we'll see you next time. Bye, everyone.